My talk will be about proteomics, as Lena just said, and the title is Leveling the Playing Field, How Post-Processing Harmonizes Peptide Identification Across Proteomics Search Engines. And as many of you are probably not from the field of proteomics or did not work that much with it yet, I will give you a very short introduction about where we are and what the very basics are to understand everything in the talk. So in mass spectrometry proteomics, we obviously first need to collect our samples. We need to do some pre-processing, like breaking up the cells and getting the proteins. Then one special thing is that we digest our proteins into peptides. So we call it digestion as enzymatic cleavage. And after this, we call it bottom-up proteomics. There are other parts, but this is not a topic of the talk today. Then it's usually given on a high-performance liquid chromatography to reduce the complexity a bit. And then we measure our samples with MS, so a double tandem mass spectrometry. And these spectra we try to identify and reference back to the peptides originally in the sample. And afterwards, we have our list of peptides, and we might conclude our proteins in the sample. My talk will be about this place here where we have our peptide and spectrum identifications. A very short introduction also how the identification works. So we have our measured MS2 spectra, and we also always need a protein database, which we enrich with some decoys. These we need later on. And we put this information together into a so-called search engine. A search engine is an identification algorithm or identification tool. And this tool creates theoretical spectra from our peptides in the database and compares them to the actual measured spectra and gives it this comparison a score. And depending on how good the score is, we say this is a good match and we trust it, and this peptide was in our sample, or we say just, this is not good, this is random, and we discard this information. It's a bit like the matching of sequence runs to a genome, if you want to compare this. So after this search engine run, we have a long list of peptide spectrum matches. And we obviously need to sort and filter this a bit and make a false discovery or Q-value filtering to only retain our good identifications. How we do this FDR estimation, this false discovery rate estimation, is that we use the decoys, which are sequences in our database which definitely are wrong identifications. And we have some ways, or some levels of how we do this estimation. There's the basic target decoy approach, which just use raw peptide spectrum match scoring. This works with only one score of our search engine. A search engine might return several metrics and several scores, but this uses only one. And these scores obviously differ between the search engines. The good thing of this approach is it is very easy to implement. You can just do it in Excel if you like. Usually you should use a tool, obviously, but it's really easy to do this. One approach which improved this very much was a so-called Percolator algorithm. This is an iterative machine learning approach which can use several of our peptide spectrometry features and scores and performs a so-called rescoring. And this was recently advanced as well with so-called with the usage of predicted spectra. And this is now the prediction-based rescoring. This creates even some more peptide spectrometry metrics and can then increase the Percolator results. For this, we have tools like MS2 Rescore and Oktoberfest, which I will also explain later. First, also a very brief overview of how the FDR estimation for the PSM scoring is done when we have only the raw scores. So we have our wrong decoy protein sequences, which can just be reversed or shuffled original sequences. and we definitely know they are wrong. If we put this together with our search engine and our spectra into the algorithm, and we get our list of peptides, we sort this by one of our scores. And as I said, we know when we have a decoy identification. So this is highlighted by this little D here now in this mockup list. And all the other identifications are true identifications. And you see we just sorted by the so-called X score. We also count how many peptides we have, decoy identifications we have up to this level. So we sort them by score, have a position in our list, and we count how many decoys we have. If we have a decoy, it gets increased, like you see here. And this can then just be used to estimate the false discovery rate with a very simple formula. And if we reach a certain position in our list where we usually have like 1% of decoy identifications, we cut this list and say, okay, we are happy with all the results above. They are good, and everything below we just discard because we are getting too much noise, too much bad identification. So this was the easy approach. Then Percolator was invented almost 20 years ago now. And here we also use this approach first to have a first estimation of our FDR, of our Q value. And then we take all of our decoy identifications, which we already have labeled as decoy, and we take only a portion of the best of our identifications, like up to 1% usually, but this can be set. Then we train an SVM, a support vector machine, for regression, which should differentiate between these good identifications and the decoys. So basically, it should learn what good identifications look like and what bad identifications look like, using all of our features we want to use, like all the scores and maybe the weight of the peptide and things like this. Then we use this trained SVM to rescore all of the PSMs using a new score, the so-called Percolator score. And based on this score, we again estimate our FDR, and this is iteratively done either until we don't have any more improvement or we have a certain level of iterations reached. So this works very well and already gives us more good identifications, because we learned what good identifications looked like, not only using the one score, but more information of our peptide spectrum matches. And then nowadays, we also have so-called fragment spectra predictions at our hands. So as I said, the search engine just uses a so-called theoretical spectrum to calculate the score. And this can be really easily calculated because our peptides or the spectra are created in a really predictable way, at least where a peak can occur. Unfortunately, we cannot easily predict the abundance or the intensity of a peak. So that's why I draw this spectra with peaks all at the same level. And this is used and compared to get our normal score. But now we have with machine learning and AI tools, we can also predict the abundance of our peaks in the spectra. And this is called a predicted spectrum then, not only a theoretical anymore. And this can now also be used to be compared against our measured spectrum. And with this, we can calculate some more metrics. This is computational and time intensive. It costs a bit, it's expensive basically, and can then be used also as an input to Percolator, these new scores. And it even improves our rescoring and gives us even more good identifications. So currently we have basically lots of search engines for DDA. This is a specific proteomics method. And all obviously are a bit different, especially when it comes to configuration and the scoring function. So the scoring function is the main difference for our identification algorithm for our search engines. And right now we have like a lot of search engines out there, These 7 are just a broad overview of the last 20 years. Some of them are really old, like X-Tandem. Some of them are really new, like Sage and MS-Fragger. And these are also the tools I used in the study I will show you now. And the question of the study was, does it actually still matter which search engine we pick, especially when we are also applying prediction-based rescoring? Is this still important, or can we get the same level of identification? And for this, we first started to create a workflow called MS-Peptide. This is a Nextflow workflow, which is migrating now into NF-Core, which you might know for sequence analysis. So also proteomics is in there now. And all of the steps are containerized as Docker images. So if you know Nextflow or NF-Core, we usually have all our tools as images, so we can run them everywhere. And as an input, we can use the raw files from vendor machines, from Thermo and from Bruker, raw files and DIF folders. We also need a faster database or a protein database, and obviously the search parameters. And the first step is that we convert these files into a standard format, which is MCML in proteomics. So we don't use the vendor formats anymore, but the standard format. We can use so-called entrapment, which I will tell you later a bit more about, and we also create a target decoy concatenation of the faster database. So we create our decoys for FDR estimation. Then the workflow has 7 search engines in there, which I just showed you. They all are a bit different and give us different results. And afterwards, we can perform some rescoring or FDR estimation, either using the default target decoy approach, Percolator, MS2Rescore, or Oktoberfest. And in the end, we get some TSV files and some plots to show us the differences between these search engines. This was benchmarked on 4 datasets, 4 public datasets available in repositories. The first was a cancer array, which had, of which we just took 10 runs of one cancer type. This total dataset had like 1,220 cancer cases measured, but we only took out 10. And this is obviously a human dataset, a medical dataset. Then we also had CAMPI, which is a metaproteomics benchmarking dataset. So there they analyzed gut or stool samples, which were synthetically created. So, but it's a special case. It's metaproteomics, so it's not human anymore, but anything which lives in this environment. And then we also had the so-called arbitrary timsTOF dataset. This is part of a dataset which was created exclusively for benchmarking, but of this we only took the DDA human samples, which is 3 runs per sample, and Orbitrap and timsTOF are mass spectrometers. So these are 2 different mass spectrometers. And as I said, everything is DDA, data dependent acquisition. This is one of the main methods used in proteomics for data acquisition. So for the cancer array, we have 10 runs. CAMPI, we only have 1 run. Orbitrap, we have 3 runs, and timsTOF, 3 runs as well. And this spans over various machines of various mass spectrometers, for which we also have some different machine parameters. The second thing we needed were the databases. For this, for the human data sets, which were the cancer, Orbitrap, and timsTOF, we took the UniProt SwissProt dataset, so human SwissProt part, together with the isoforms, which were then almost 43,000 protein entries. It's more than these 20,000 genes because of the isoforms. The human reference proteome, which is even a bit bigger and contains some more truncated and even more isoforms of these proteins. And we also took the so-called ProHub, which is a haplotype database. So it carries lots of variants of haplotypes in the environment, which then is already 350,000 proteins which has 350,000 proteins in there. For the CAMPI dataset, we use the provided CAMPI databases, which were two. One is the IGC reference catalog, which contains almost 10 million proteins in there, which is from different species and which is derived from a transcriptomics and genomics sequencing approach of gut samples. So it's the proteins obtained from there. So it's multi-species and lots of different proteins or possible proteins. And we also have a smaller one, which is the DB2 then, which only has 442,000 proteins in there, which was a bit of a they took some special samples on their own again and created just a smaller database. And we always added some contaminants which just might occur during proteomics samples. So then we performed all these runs, all these searches. 3 or 2 databases per dataset, 7 search engines, and our rescoring algorithms, and we looked at the results. So first, just an overview, we had a timsTOF dataset here, we had the SwissProt database, the different post-processing steps, the raw search results, the basic target decoy approach, then Percolator, MS2 rescore, and Oktoberfest. And what is shown here is just the numbers for each search engine of returned peptidoforms, which is basically peptides with modifications. And you see there's some difference in numbers. Some of the search engines did not perform really good with some post-processing, like X-tandem here. I will come back to this later. MaxQuant somehow obtained the same level, but with Percolator, MS2 rescore, and Oktoberfest, we seem to have increased our identifications. This is looks more or less the same if you use the human proteome database, so not much of an increase there, even if this database is larger. And for the ProHub, something looked very different. So suddenly for the MSGF Plus results with MS2 rescore and Oktoberfest, we had like 4 times of the number of identifications. So this is valid FDR 1% identifications of peptidoforms. So this looks really weird and cannot be right. And to figure out what actually happened there, we looked at our target and decoy distributions. So we know that the FDR estimation methods always assume a certain target decoy distribution, which means that the decoys should have similar distributions as wrong target identifications. And this might be something which just went off here. So to check this, we looked at these distributions and we created histogram plots where we plotted just the number of identifications against the score of the search engine. And here we see this for Comet and MSGF+. In green are our target identifications, and in orange our decoy identifications. And you see really nicely here that for both of them, the decoys follow the same curve as our targets in a low scoring region. So we can assume that these follow bad identifications or wrong identifications as they should. When we perform Percolator, we see 2 things. First, we have a better separation between good and bad identifications. So we see basically 2 hills here. And we still see that with Comet, the decoys really nicely follow this bad identification distribution. But for MSGF+, the separation looks a bit off. It follows the same trend, but the scores are a bit lower. And if you do this further, for MS2 rescore and Oktoberfest results and Comet, we see again the separation even got better, and the decoys still follow this the low scoring targets. But for some reason on MSGF+, suddenly the decoys are not on the same distribution anymore, but yeah, the algorithm learned to discern between targets and decoys, which we definitely don't want. We can do this on our own, but this is not what we want to see. It still should follow the same distribution. This luckily could be fixed by removing one of the MSGF plus scores for the rescoring. So MSGF plus already had a score in there which discerned too well between targets and decoys and not only between good and bad identifications. With removing this, we fixed this, and the MSGF plus distributions look like the comet distributions. Afterwards, we checked this as well, and there were no errors left. But just to show the impact, we kept the Percolator, the MSGF plus scores for Percolators still there. So this slight shift here might still be there for the rest of the analysis. Okay, so after we fixed this, we looked at our identification rates again, showing here for the Orbitrap dataset. And what is shown here is again the number of peptidoforms, of identified peptidoforms, plotted against the FDR value for all of the search engines. And we see a trend that which is seen in all of our data sets and all of our databases, that with the raw search results, the separation between the pure numbers is still really different. With Percolator, it is squashed a bit together, and MS2rescore and Oktoberfest squashes this even more together. So it looks already like there's some better consensus if you only look at the number of identifications. This holds for all of, basically all of our combinations. And let's look at this a bit more in depth for the cancer array dataset, the human proteome database. So what we see here now is that we have a large increase again of consensus of identifications when we perform Percolator, MS2rescore, or Oktoberfest in comparison to the raw search results. And we calculated here the differences between the maximum versus the minimum of our identifications. So, for example, on the raw research search results, the number of SAGE identifications against the X-tandem identifications in percentage. And this was like almost 67% or 15,000 identifications difference for the raw search results. Just if you wonder why we have these error bars here, it's because we have this the mean of these 10 cancer runs which we analyzed. So this difference is quite high still. For Percolator, this shifts to only 7,000 different identifications. We left out MaxQuant here because it did not improve that much. And for MS2rescore and Oktoberfest, these differences go down to almost only 2,000 peptides in difference, or below 10% difference between all the search engines, which is really nice to see. This is similar for all of our data sets and machines, databases. And there are also some really interesting trends, like this is what I showed you before, the timsTOF dataset with the proteome. And what you see here is that with raw search results only, X-tandem had really low identifications. So this is the FDR valid or FDR 1% identifications below an FDR of 1%. And this is really low because for some reason X-tandem scored the decoys really high there. But with using Percolator, this is pretty much rescued, even more with MS2rescore or Oktoberfest. So this really gives you a large gain and homogenizes the identifications again. For the CAMPI dataset, we also have more or less the same trends. The only very interesting difference here is that MaxQuant actually performed much better it was also much increased by the rescoring, and MS Amanda did not perform as good as before. If you see MS Amanda in the other plots, it was almost on par with the other search engines, and here it was a bit lagging behind. But this might just be slight tendencies for this very big metaproteomics data set. So after we saw this, we wanted also to see how big actually the impact of different databases were. So what is shown here is only the results for SAGE. And we performed some upset plots where we can show how big the overlap between the data sets, post-processing, and the results is. So basically what we see here is for SAGE on the Kansai array, the total overlap is relatively large, which we expected. And the biggest increase we see is actually when as soon as we perform rescoring, like Oktoberfest MS2 rescore, rescoring. So this big overlap here is the largest increase we see. So rescoring really is the thing which gives us a more consensus in the identifications and a big increase in the overlap. This can also be seen for the Orbitrap and the timsTOF dataset. For the timsTOF, not as much actually, but for the Orbitrap, about the same range. Very interesting. On the CAMPI dataset, this looks a bit different because here we get the highest increase when we actually change the database. So the database 2 is shown here, and as soon as we introduce the database 1 or change to database 1, we get much more identifications, also overlapping, so consensus identification. This is even more increased with the rescoring, but the biggest increase is the database. And this somehow shows us that the database 2 might not be comprehensive enough. It might just not cover all the species in our metaproteomics analysis. To get a larger picture of this, this plot was created, which just shows us the overlap on peptidoform level for each of the combinations of data search engines for each database and each data set. So these 3,611 peptidoforms were identified by all the 7 search engines using SwissProt and the Kansai array and so on. And up here we showed the highest identification for one search engine in this combination of database and data set. So what we again see here really nicely is that for the raw PSM scores, the overlap is still relatively small, especially if you have something like the X tandem, which had some scored some decoys really high, so that the FDR valid peptidoforms were relatively low. And Percolator already increases this a bit, or also a lot in some of the samples like here. But the prediction-based rescoring really is the big increase. So we really see that we have many more consensus identifications between our search engines. And this is up to 3-fold in cases like here, or even up to 10-fold increase if we look at this badly behaving data set. So this was really nice to see that this rescoring really has a big impact on making our identifications more comparable. But one last thing we should check was how does our false discovery rate estimation actually behave? So can we trust our FDR estimation? And for this, just recently, a method was introduced and analyzed, which is the so-called entrapment strategy. And for this, we also created a 3-fold shuffled entrapment database for each data set. And this allows us the estimation of the so-called false detection probability. So basically, it is another level of wrong identifications in our database, which allows us to estimate how well the false discovery rate estimation works against definitely wrong identifications again. The only thing we can do with this is to calculate the upper and lower bound of our false detection probability. For something more exact, we needed to have some other databases, which we did not create here. And the plots I show you will look a bit like these. Sorry, they are at the first a bit hard to read, but I will quickly show you how they are meant. So we always have the lower bound, like here in blue, the lower bound for our raw PSM searches only, and the upper bound for our raw PSM searches only in a dark blue shape. It's again, it's like a tunnel because we have multiple runs aggregated there. And our actual FDR estimation should not be above our upper bound of the FDP estimation. So basically, this diagonal, which is drawn here when we plot the FDR-Q value against the FDP, should not go be above our upper bound, which actually holds true for these raw searches. It also more or less holds true if we use Percolator. So it, as you see, this dark green lines are a bit above the FDP, but not too much. And for MS2 rescore, which is this orange brownish shape, or Oktoberfest, which is this gray, the upper bound is already a bit above what we want it to be. But for most of the combinations, it actually looks okay-ish, and the FDP is at least not below 2% for an FDR of 1%. So if you look here, here's the FDR 1% limit. As I said, for raw PSM scores and Percolator, it looks good anyways. But for MS2 rescore and Oktoberfest, it goes to 1.3% maybe. So it's still okay-ish. But there were some very interesting cases. First, we look at MS Amanda on the Orbitrap dataset. And there we see something like this, where each combination is well below the FDP, the estimated FDP. And this also goes on for the whole FDR Q value range or FDP range. So it even stays lower if we increase the FDR values. This shows us that MS Amanda is rather conservative, which is not bad per se, but we might even lose some good identifications with this behavior. So we say we allow up to 1% bad identifications and hopefully also the good identifications. But if we are too conservative, we might even lose some of the good identifications. So, but still, this is okay. Something really weird was this behavior, which we saw on the MS Fragger for the Orbitrap. So basically, all of the combinations are more or less okay, but Oktoberfest has a really interesting behavior there. As you see, it really underestimates the FDR over the whole range shown here, and this goes on for the for much larger FDR Q values as well. So the upper limit of the estimated FDP comes back to the FDR value, but at ranges of like 14% FDR, which you would never allow in your sample. And the lower bound goes there much quicker, and this behavior somehow looks interesting. It looks like somehow the good identification, the really true identifications were just too good for this adjustment maybe. So the Oktoberfest guys are already on trying to explain this behavior better and fixing their algorithms or getting another fix for the entrapment. As I said, this goes back to normal behavior, but on a really high level, which you would never allow in your analysis. So let's finish with some practical considerations. Because actually your choice of search engine might also be I'd rather be on the resources and time restrictions you have, and also a bit on the pipeline you need to use. So we also tracked how much time and how much CPU and memory we actually consumed for these analyses. And this is just shown for the Orbitrap dataset here. And what you see here is how long one part of the analysis took and how many instances we actually needed to analyze the whole data set, which then uses this, this time and CPU. So if you look, for example, at Comet, one Comet run took like 240 seconds to run, and it used 16 CPUs and a peak memory of below 6 gigabytes. This even could scale. You can run them in parallel. If you have this amount of memory CPUs, then you can finish it still in the same time. Max Quant took much longer and used also much more memory. And MSGF Plus, there we needed to split the samples into several into smaller chunks, and each of these chunks took almost almost 50 minutes to process, which is a bit long. It is not really parallelizable anymore. But if you need to choose your search engine, you really should or might want to keep this in mind as well. So Comet, relatively quick. MS-Fragger and Sage were intended to be quick as well. Depends a bit on a data set here, but maybe this is also needs to be taken into consideration if you want to choose your pipeline. So this is my final slide, the conclusions and take-home messages. So what we showed in this study is that the prediction-based rescoring really harmonizes the identifications across the engines. So basically saying it does not really matter anymore which search engine you use, as long as you use prediction-based rescoring right now, because the results, they get the same, unless for some very specific cases I showed. The bottleneck actually in the search engines is also the scoring power and not the candidate generations, because we also showed that the overlap of our identifications is really good after the rescoring. So the candidates, the PSMs are actually there. Only we cannot discern between bad and good identifications enough. So that's the problem in search engines right now. The database matters when it is very incomplete, like for metaproteomics samples. But on human samples, it basically does not matter that much. So if you use the human reference proteome, that's fine. If you want to look for variants and need a much larger database, that's fine as well. We might be able to find them there, but we don't lose much of our basic identifications. You should always validate the FDR on your workflow and use entrapment. This should only be done for a small part of and to check your workflow and not on the whole data set. But if you have the time, it's a good choice to do this. And probably the choice of your search engine is rather driven by compute and general pipeline needs. What was not done here was that we did not look at some interesting biological modifications. So we just took the basic ones, oxidation of methionine and carbamidomethylation of cysteine, but nothing too interesting. This might behave a bit different. And we only looked at tryptic peptides here. So this might also change a bit if we have some other special methods. The pipeline used MS-PepID is in NF Core as a development version right now. If you want to look at the preprint of all of this, you can have a look there or just search for the name. With this, I come to thank some persons. Like I thank my group, which is Dirk, Sven, and Felix. And I also thank the whole core unit for bioinformatics, which is headed by me and Martin Eisenacher, and obviously also the colleagues there. And I have one short slide of advertisement. If you are working with mass spectrometry does not need to be proteomics, can also be metabolomics or something else. Would be nice if you have a look at this UBIC hackathon we are organizing right now. If you have an idea for a hackathon or for a project, you can submit it, or just be part of the community and work for 1 week on really nice topics. Okay, with this, I thank you for your attention, and I'm free to take some questions.